Now we'll start discussing about the section C, that is long answer questions. You have 3 questions, 19th, 20th and 21 question, 3 questions, these are the numbers. So you have to answer any 2 of these questions. So the first question, that is the 19th LAQ I am going to discuss, is LAQ number 19 is explain C3 cycle. C3 cycle. We all know that this is a very important cycle. Even for that of the competitive track also it is very important. Okay, C3 cycle. Now regarding C3 cycle, we must be able to know. 3 scientists know that, 3 scientists, that is Melvin Kelvin, Andrew Benson and James Basham. Okay, Melvin Kelvin, Andrew Benson and James Basham are the 3 scientists, or you can also say Melvin Kelvin is the main scientist and his associates Andrew Benson and James Basham worked out on their experimental material was chlorella. Worked out on chlorella and have discovered this pathway, that is C3 pathway. So the 3 scientists, Melvin Kelvin, Andrew Benson, James Basham, 3 of these workers, the experimental material was chlorella. They have discovered C3 pathway in this chlorella. And here, why is it named as C3 pathway? It is named as C3 pathway because the first product which was formed of the end product of the first step of the reaction which was formed was a 3-carbon compound. As the end product of the first reaction which was formed was a 3-carbon compound, the pathway is named as, what is it named as? C3 pathway. It is named as C3 pathway. Okay, so that's why the C3 name has been come basing upon the first end product of the first reaction, end product of the first step of the reaction. That's what. And it is also called as Calvin cycle. It is also called as Calvin cycle. The name Calvin cycle has come in the honor of the scientist, that is Melvin Kelvin. Melvin Kelvin for his this conspicuous work which has been done, he received a Nobel Prize in the year 1961. Melvin Kelvin received Nobel Prize in the year 1961. So it is called as, that's why C3 cycle, it is also called as Calvin cycle in the honor of the scientist Melvin Kelvin. So one name is C3 cycle and the other name is Calvin cycle. And what are the other names? Other names are RPP pathway. RPP pathway is reductive pentose phosphate pathway. RPP pathway is reductive pentose phosphate pathway, and it is also called as PCR pathway. PCR pathway full form is photosynthetic. It is photosynthetic carbon reduction pathway. So these are the names of the C3 cycle. C3 cycle, Calvin cycle, reductive pentose phosphate pathway, PCR pathway, that is photosynthetic carbon reduction pathway, which was carried out in chlorella by the 3 scientists. Okay, so this C3 cycle can be C3 cycle, it occurs in three steps, under three steps. What are the three steps? One is carboxylation. The second one is reduction, carboxylation, reduction, and the third one is regeneration. Okay, carboxylation, reduction, and regeneration. Here the carboxylation or Calvin cycle starts with that of the carboxylation step. What is carboxylation? Here carbon dioxide is absorbed, or rather it is accepted from the atmosphere by a primary carbon dioxide acceptor. Okay, for example here 6 molecules of carbon dioxide are accepted by a primary carbon dioxide acceptor, we are telling. The primary carbon dioxide acceptor here is 6 molecules of ribulose 1,5-bisphosphate, in the presence of an enzyme Rubisco. The enzyme is here enzyme Rubisco, which results in the formation of 12 molecules of 3-PGA, that is phosphoglyceric acid, which is a 3-carbon compound. Ribulose 5-bisphosphate is a 5-carbon compound. This is the first step, actually we can say. Okay, so carboxylation is the first step under C3 cycle, wherein it starts with that of absorption of the carbon dioxide from the atmosphere by a primary carbon dioxide acceptor. Here, for 6 molecules of carbon dioxide to be absorbed from the atmosphere, it requires 6 molecules of primary carbon dioxide acceptor. Here the primary carbon dioxide acceptor is ribulose 1,5-bisphosphate, which is a 5-carbon compound. The carboxylation step is activated, or rather it is carried out by an enzyme, that is Rubisco. What's the full form of Rubisco? Ribulose 1,5-bisphosphate carboxylase or oxygenase. Carboxylase or oxygenase. Okay, which results in the formation of 12 molecules of 3-phosphoglyceric acid, which is an 3-carbon compound. Then now coming to that of the speciality or the significance of this enzyme Rubisco. Rubisco means what I have said, that is ribulose 1,5-bisphosphate carboxylase or oxygenase. Why are we not restricting to one? Because the enzyme Rubisco has a dual nature. What is this dual nature? Dual nature means it is going to have two natures. What are the two natures? The nature, or we can say the activity of the Rubisco is decided basing upon the volume, or rather the percentage of the oxygen which is present in the atmosphere. For example, we all know that the general percentage of carbon dioxide which is present in by volume, it is by volume, in the atmosphere is 0.03% by volume of carbon dioxide is present in the atmosphere. Then coming to that of oxygen. Okay, oxygen is present in 21% by volume in the atmosphere. If at all the percentage of oxygen is going to exceed beyond 21 by volume in the atmosphere, then the same enzyme Rubisco is going to behave as an oxygenase, and it is going to carry out a waste process called as C2 cycle, or rather it is commonly called as photorespiration. It is called as photorespiration. respiration. So that's what the nature of the rubisco is mainly decided basing upon the percentage of oxygen which is present in the atmosphere. If at all the oxygen is 21 and below 21, then rubisco enzyme is going to carry out carboxylation step and it is going to carry out this reaction, carboxylation. Whereas if it is beyond 21%, the percentage of oxygen is going to be on 21%, then it is going to carry out a waste process called as photorespiration, which is also called as C2 cycle. So that's what. So what is the first product which is formed? 12 molecules of 3-phosphoglyceric acid, which is a 3-carbon compound. As the first compound which is formed is an 3-carbon compound, that's why the pathway is named as C3 cycle. So this is the first step, that is carboxylation step. Now coming to that of the next step, that is reduction. The next step, that is reduction. Okay, so the product which is formed is 3PJ. That now 12 molecules of 3-phosphoglyceric acid, which is a 3-carbon compound, it is reduced. It is reduced with 12 molecules of ATP in presence of an enzyme. In presence of an enzyme, that is phosphoglycerokinase. In presence of an enzyme phosphoglycerokinase, which results in the formation of 12 molecules of 1,3-bisphosphoglyceric acid. 12 molecules of 1,3-bisphosphoglyceric acid plus 12 molecules of ADP. 12 molecules of ADP. 12 molecules of ADP plus 12 PI, inorganic phosphate. This is the reduction step. Here, the first product which is formed as a result of carboxylation, that is phosphoglyceric acid. 12 molecules of phosphoglyceric acid undergoes reduction with 12 molecules of ATP. ATP is nothing but adenosine triphosphate. Where do you get this ATP? ATP is the light generated ATP. That means which has been, ATP is nothing but the assimilatory power which has been generated during the light reaction. Okay, so in the presence of an enzyme, this reduction step is carried out in the presence of an enzyme phosphoglycerokinase. Phosphoglycerokinase, resulting in the formation of 12 molecules of 1,3-bisphosphoglyceric acid, wherein which the byproducts like 12 molecules of ADP, adenosine diphosphate plus 12 PI, inorganic phosphate. This is the second step of reduction. Now, this 12 molecules of 1,3-bisphosphoglyceric acid are further reduced. These are further reduced. 12 molecules of 1,3-bisphosphoglyceric acid are further reduced with another assimilatory power, that is 12 molecules of NADPH plus H plus in the presence of an enzyme. What is the enzyme over here? Catalyzing this reaction, glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase is the enzyme which catalyzes the second step of reduction, wherein it results in the formation of 12 molecules of glyceraldehyde 3-phosphate plus 12 molecules of glyceraldehyde 3-phosphate plus 12 molecules of NADP plus, here NADPH, which is oxidized to which is reduced to, sorry, it is oxidized to NADP plus 12 molecules of H2PO4 minus. 12 molecules of H2PO4 minus. Okay, so here 12 molecules of 1,3-bis phosphoglyceric acid is reduced in presence of 12 molecules of another assimilatory part, that is NADPH, in the presence of an enzyme glyceraldehyde 3-phosphate dehydrogenase, results in the formation of 12 molecules of glyceraldehyde 3-phosphate plus 12 molecules of NADP plus 12 molecules of H2PO4 minus. So, these are the two steps under the reduction, that is the second step under C3 cycle. Okay. So, under C3 cycle, the second step of the C3 cycle, the product which is formed, that is 3-phosphoglyceric acid, undergoes reduction. First it is undergoes reduction with ATP in the presence of an enzyme phosphoglycerokinase, resulting in the formation of 1,3-bis phosphoglyceric acid. Wherein this 1,3-bis phosphoglyceric acid, it further undergoes reduction in the presence of 12 molecules of NADPH. It is also an light generated assimilatory part, which results in the formation of 12 molecules of G3P, glyceraldehyde 3-phosphate. Now, the resultant which is formed at the end of the reduction step, the resultant which is formed at the end of okay, the resultant which is formed at the end of reduction step is, that is nothing but G3P pool, you can say, that is 12 molecules of glyceraldehyde 3-phosphate. It is called as G3P pool. So, first we must be able to know what happens to these 12 molecules of G3P pool. Here, out of these 12 molecules, we can simply say that, so out of these 12 molecules of G3P pool, 2 molecules of G3P are exported out to cytosol, What for they are exported out to cytosol? Cytosol is nothing but cytoplasm. So, these are exported out to cytosol for the synthesis of hexose or the glucose molecule, So, through this, it is very clear that out of the 12 molecules of G3P pool, 2 molecules are exported out to the cytosol, that is nothing but cytoplasm, wherein they are synthesized to what? Hexose molecules, hexose. So, that is why here it is very clear that synthesis of hexose takes place in the cytoplasm. Here, and you can also say the end products of C3 cycle. Actually, the end products of C3 cycle. The end products of C3 cycle are nothing but disaccharides, sugars, starch, in any form of this, you can say. Okay. So, here the end products or the actual synthesis of the sugars takes place in the cytoplasm, wherein in any other form the end products of the C3 cycle are disaccharides, sugars and starch, and these end products either they are stored in the form of starch and usually they are stored in which tissue? That is a phloem, where these end products of C3 cycle are stored in the form of starch, or rather they are used up in the phloem cells. So that's what we can say that. So the end products of C3 cycle are disaccharides, sugars and hexose molecules, whatever they are. Okay, so out of 12, 2 molecules are exported for this synthesis, and actual synthesis of hexose takes place in case of the, or in which region? That is in case of the cytoplasm, which can also be called as cytosol. Okay, now what happens to the rest of the 10? Now, as out of 12 molecules of G3P, 2 molecules are exported out to the cytosol for the synthesis of the hexose, whereas the rest of the 10 molecules enters into a process called as regeneration. Regeneration of what? Here, regeneration of the primary carbon dioxide acceptor. Here the primary carbon dioxide acceptor is ribulose 1,5-bisphosphate. That's what it takes place. So out of the 12, as the 2 molecules of G3P are utilized for the synthesis of hexose, 10 enter into a next step called as regeneration, wherein primary carbon dioxide acceptor, that is ribulose 1,5-bisphosphate, are regenerated in order to continue the pathway, or in order to continue the cycle. So here I am now going to draw the cycle. The C3 pathway. Come, I am going to draw, now show you the C3 pathway, that is wherein 6 molecules of CO2, these are absorbed by a primary carbon dioxide acceptor, that is ribulose 1,5-bisphosphate, 6 molecules of ribulose 1,5-bisphosphate in the presence of an enzyme, Rubisco. Which results in the formation of 12 molecules of 3-PGA. 12 molecules of 3-PGA, that is 3-phosphoglyceric acid. So that's what. In order to take 6 molecules of carbon dioxide from the atmosphere, it requires 6 molecules of ribulose 1,5-bisphosphate, which is an primary carbon dioxide acceptor in the presence of an enzyme ribulose 1,5-bisphosphate carboxylase or oxygenase. Here one thing you must remember is that Rubisco is an enzyme which accounts for about, remember this point, which accounts for about 25 to 50% of the total soluble proteins which are present in the leaf. Okay. Here regarding the enzyme Rubisco, that is ribulose 1,5-bisphosphate carboxylase or oxygenase, it accounts for about 25 to 50% of soluble proteins, 25 to 50% of the total soluble proteins present in the leaf. This is one of the significant feature of the Rubisco, as I have already discussed that Rubisco, which has a dual nature basing upon the percentage of the oxygen in the atmosphere, that is one character you can take into consideration, and the other one is that it is the most important or most abundant available protein in the world. It is the most abundant protein in the world. That is one more point. And one more point is that it accounts for about 25 to 30% of the total soluble proteins present in the leaf. So that's what about the enzyme rubisco. So due to the activity of this enzyme rubisco, which results in the formation of 12 molecules of 3-phosphoglyceric acid, which undergoes reduction. Reduction by what? By 12 molecules of ATP, resulting in the formation of ADP plus 12 pi, which results in the formation of 12 molecules of 1,3-bis-phosphoglyceric acid. This reduction is catalyzed or rather carried out by an enzyme phosphoglycerokinase. By an enzyme phosphoglycerokinase. Now this 12 molecules of phosphoglyceric acid, bis-phosphoglyceric acid, 1,3-bis-phosphoglyceric acid, 1,3-bis-phosphoglyceric acid, it undergoes further reduction. In by 12 molecules of NADPH plus H plus, which results in 12 molecules of NADP plus 12 molecules of H2PO4 minus. Okay. In the presence of an enzyme, what is the enzyme catalyzing this reaction? Glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase is the enzyme catalyzing this step. Now, which results in the formation of 12 molecules of glyceraldehyde 3-phosphate. 12 molecules of glyceraldehyde 3-phosphate. So this what? This is the first step, that is carboxylation step, and these are the two steps. These are the two steps of reduction. Okay, so that's what, this is the carboxylation step and this is the reduction step. So due to the space area, I am going to rub this carboxylation as well as reduction, and we will discuss about the G3P pool happenings. What are going to happen regarding the G3P pool? Okay. Now coming to that of the 12 molecules of G3P. So these are called as G3P pool, as I have said you. G3P 1, 2, okay, so 2 molecules of each we can go with, it will be easy for us. Okay, so 1, 2, 3, 4, 5, 6, 6 to 12. So total 12 molecules of G3P which are produced as a result of reduction by using the assimilatory power which has been generated from the light reaction. This is called as G3P pool. Out of this, 2 molecules of G3P are, these are exported out. These are exported out to cytosol. Cytosol for synthesis of hexose molecule. These are exported out to cytosol for the synthesis of hexose. That's what. So out of 12, 2 has been exported out for the synthesis of hexose in the cytosol. Now coming to that of the rest of the 10 molecules of G3P. Out of this 10 molecules of G3P, what happens now? Okay. Out of this 10 molecules of G3P, 4 of the G3P molecules, they are isomerized. 4 are isomerized. Isomerization is nothing but intramolecular shift. Isomerized to, they are isomerized to DHAP. DHAP is dihydroxyacetone phosphate. So, 4 molecules of glyceraldehyde 3-phosphate are isomerized to DHAP, that is dihydroxyacetone phosphate. Dihydroxyacetone phosphate. Okay. So, here what happens over here? In the present, this isomerization is carried out in the presence of an enzyme triose phosphate isomerase. It is carried out by an enzyme triose phosphate isomerase. Okay, now two molecules of G3P combines with these 2 molecules of DHAP. 2 molecules of G3P combines with 2 molecules of G3P combines with 2 molecules of DHAP. Combines with 2 molecules of DHAP, and this reaction is catalyzed by an enzyme aldolase. And the step over here, remember, the step over here is combining or combination, you can go with. Okay, so here 2 molecules of G3P combines with 2 molecules of DHAP in the presence of an enzyme aldolase, which results in the formation of fructose 1,6-bis phosphate. Fructose 1,6-bis phosphate. This fructose 1,6-bis phosphate, it undergoes dephosphorylation. That is, it loses inorganic phosphate in presence of an enzyme. In presence of an enzyme, fructose 1,6-bis phosphate, fructose 1,6-bis phosphatase. Okay, so here what is the step carried out over here? It is dephosphorylation step. This is the dephosphorylation step. Okay, so fructose 1,6-bis phosphatase is the enzyme which is going to carry out this dephosphorylation reaction, which results in the formation of fructose 6-phosphate. Now, this fructose 6-phosphate, again the same 2 molecules you can go with. Now, this fructose 6-phosphate, it combines. Fructose 6-phosphate, now it combines, again combines with 2 molecules of G3P, which results in the formation of 2 compounds over here, and this reaction is catalyzed by an enzyme that is transketolase. Okay, so 2 molecules of fructose 6-phosphate combines with 2 molecules of glyceraldehyde 3-phosphate in presence of an enzyme transketolase. Results in the formation of erythrose 4-phosphate and 2 molecules of erythrose phosphate and 2 molecules of xylulose 5, 2 molecules of xylulose 5-phosphate. Okay, now these 2 molecules of erythrose 4-phosphate, they both combines. Here again combining is the step. Combines two molecules of DHAP. Here the enzyme catalyzing this combining step is aldolase. Okay, wherein it results in the formation of pseudoheptulose 1,7-bis phosphate. Pseudoheptulose 1,7-bis phosphate. Two molecules again over here. This pseudoheptulose 1,7-bis phosphate, it undergoes again dephosphorylation. Dephosphorylation results in the formation of pseudoheptulose 7-phosphate. Pseudoheptulose 7-phosphate. This pseudoheptulose 7-phosphate, now it combines, and here this dephosphorylation step is carried out by pseudoheptulose 1,7-bis phosphatase. This is the enzyme which catalyzes the reaction, and here the pseudoheptulose 7-phosphate, which is formed after dephosphorylation, combines, again combining, okay, in presence of an enzyme that is transketolase. In presence of an enzyme that is transketolase, which results here, we can, lack of space, I will be writing over here. Okay, so transketolase, which results in the formation of two compounds, that is, one is two molecules of ribose 5-phosphate and two molecules of again cellulose 5-phosphate. Cellulose 5-phosphate. Now this total, now how many? 4 molecules of cellulose 5-phosphate are formed over here. Now these 4, this one over here as well as these. Now these 4 molecules of cellulose 5-phosphate, they undergo 4 molecules of cellulose 5-phosphate, they are converted, they undergo conversion. They are converted to 4 molecules of ribulose 5-phosphate, and this reaction is carried out by an enzyme ribulose, that is ribulose 5-phosphate epimerase, ribulose 5-phosphate epimerase. Okay, meanwhile this another molecule which is formed over here, that is ribose 5-phosphate, it undergoes isomerization. It undergoes isomerization in the presence of an enzyme, that is ribose 5-phosphate isomerase. Ribose 5-phosphate isomerase, which results in the formation of 2 molecules of ribulose 5-phosphate. Ribulose 5-phosphate. Now total 6 molecules of ribulose 5-phosphate are formed. These 6 molecules of ribulose 5-phosphate, they are phosphorylated with 6 molecules of ATP. Here for the phosphorylation step again 6 molecules of ATP are utilized. 6 molecules of ATP are utilized, which results in the formation of 6 molecules of ribulose 1,5-bis phosphate, which is a primary carbon dioxide acceptor. Actually this step has to come over here so that it carries out the carboxylation step. And this entire cycle over here, which is presented on the board, shows about the regeneration process, shows about the regeneration step, that is the third step under C3 cycle. Okay, as we have discussed that carboxylation step, as a result of carboxylation, which results in the formation of 12 molecules of 3-phosphoglyceric acid, which is a 3-carbon compound, which undergoes 2 steps of reduction, first reduced by 12 ATP molecules, later reduced by 12 NADPH molecules, finally resulting in the formation of 12 G3P molecules, which are called as G3P pool. Out of these 12 G3P molecules, 2 are exported, 12 are focused over here, the 12 G3P molecules. These are the 12 G3P molecules. So, out of these 12 G3P molecules, the 2 G3P molecules are exported out into the cytosol in order to synthesize hexose. That is why here the end product of C3, that is hexose, which are synthesized in the cytosol. Now coming to that of the rest of the G3P molecules, the 10 G3P molecules. Out of this, 4 G3P molecules are isomerized to form DHAP, that is dihydroxyacetone phosphate, in the presence of an enzyme, that is triose phosphate isomerase. Then out of this, 2 G3P molecules combines with 2 DHAP over here, which is catalyzed by an enzyme aldolase, results in the formation of 2 molecules of fructose 1,6-bisphosphate. This fructose 1,6-bisphosphate undergoes dephosphorylation in the presence of an enzyme fructose 1,6-bisphosphatase, resulting in the formation of fructose 6-phosphate. This fructose 6-phosphate combines with another 2 molecules of G3P, resulting in the formation of 2 compounds, that is erythrose 4-phosphate and cellulose 5-phosphate, in the presence of an enzyme transketolase. Now erythrose 4-phosphate, it combines with the another two DHAP molecules, resulting in this combination is carried out by an enzyme aldolase, which results in the formation of pseudoheptulose 1,7-bisphosphate. And this pseudoheptulose 1,7-bisphosphate undergoes dephosphorylation, which is catalyzed by an enzyme pseudoheptulose 1,7-bisphosphatase, resulting in the formation of pseudoheptulose 7-phosphate. This pseudoheptulose 7-phosphate combines with the last 2 molecules of G3P in the presence of an enzyme transketolase, which results in the formation of, again this combination activated by the same enzyme which has seen earlier, that is transketolase, resulted in the formation of 2 compounds, that is ribose 5-phosphate and cellulose 5-phosphate. Ribose 5-phosphate and cellulose 5-phosphate. So totally, here we can go, it is actually an 8-step process we can go with, is that this is isomerization totally of 1, okay, then combination 2, then dephosphorylation 3, then another combination, that is 4, then combination 4, then the next one is 5, another combination, then 6 dephosphorylation over here, then 7 step, that is combination over here, and coming to that of the formation, that is the here 8th step, and this is the 9th step, and this is the 10th step. Okay, 10th step is here, the 10th step is phosphorylation. Okay, so and the whole at the end we can say that total regeneration process is a 10-step process wherein we have come over here, isn't it? So, the last, that is pseudoheptalo 7 phosphate, which is formed due to dephosphorylation of pseudoheptalose 1,7 bisphosphate, resulting in the formation of two compounds, that is ribose 5 phosphate and xylulose 5 phosphate. Xylulose 5 phosphate, which is already formed over here, xylulose 5 phosphate, isn't it? That is over here in this step. Then total number of xylulose 5 phosphate which is formed are 4 molecules. These 4 molecules of xylulose 5 phosphate undergoes, or rather 4 molecules of xylulose 5 phosphate are converted to 4 molecules of ribulose 5 phosphate in the presence of an enzyme ribulose 5 phosphate epimerase. Now meanwhile, another compound which is formed, that is 2 molecules of ribose 5 phosphate, it undergoes isomerization in presence of an enzyme ribose 5 phosphate isomerase, resulting in the formation of ribulose 5 phosphate. So total in the previous step, that is in the 8th step as well as 9th step together, which resulted in the formation of 6 molecules of ribulose 5 phosphate. Now these 6 molecules of ribulose 5 phosphate are phosphorylated by utilizing 6 molecules of ATP. Again the ATP comes from the light reaction, which resulted in the formation of 6 molecules of ribulose 1,5 bisphosphate, which is a primary carbon dioxide acceptor. Now the regeneration of the primary carbon dioxide acceptor has taken place, that is 6 molecules of ribulose 1,5 bisphosphate has taken place. The phosphorylation step is catalyzed by an enzyme ribulose 1,5 bisphosphate kinase, which catalyzes the phosphorylation step. So that's what the end of the regeneration, wherein the primary carbon dioxide acceptor, that is 6 molecules of ribulose 1,5 bisphosphate has been regenerated, and now this primary carbon dioxide acceptor is ready to accept carbon dioxide from the atmosphere, or ready to fix carbon dioxide from the atmosphere. So finally, coming to the end of the C3 cycle, if you go with over here, that the energetics, how much or how many assimilatory power are utilized, over here. The assimilatory power utilized over here, first during the reduction step, okay, ATP, how many ATP is utilized during the reduction step? Total 12 molecules are utilized during reduction step, and again 6 molecules of ATP are again utilized during regeneration step. So total how many ATP molecules? 18 ATP molecules are utilized during C3 cycle in order to produce one hexose molecule, as well as not only ATP but also NADPH which was utilized. There is only NADPH which was utilized, 12, that is during reduction step. So at the end of C3 cycle, if we summarize, C3 cycle under 3 steps, that is carboxylation, reduction and regeneration. Carboxylation step, wherein you get the 3 PGA, and reduction step wherein you get 12 molecules of 1,3 bisphosphoglyceric acid, okay, whereas under regeneration step, wherein the primary carbon dioxide acceptor is regenerated, that is ribulose 1,5 bisphosphate. Okay, so in order to produce or in order to synthesize one molecule of hexose, the total number of assimilatory power which is utilized under C3 cycle is 18 ATP molecules, that is in clear if you go with 12 ATP molecules during the reduction step and 6 during the regeneration step, and 12 NADPH molecules during the reduction step. So this is what about the C3 cycle in detail. We have finished discussing about C3 cycle.